Welcome back to Nursing Hub. Today in this video, let's see a new topic, that is genetic testing. Before going into the topic, let's see what is the definition of genetics. So genetics, as we know, it's a branch of biological science which deals with the genes and heredity, or we can say it is how the transmission of characteristic which happens from parent to the offspring. The term genetics was coined by Bateson in 1906. It has been derived from the Greek word gene, that means to become. The main components of genetics are heredity and variation. So first one is heredity. So it's the study of factors responsible for the resemblance between parents and their offsprings. So sometimes we say that father and the child, father and daughter look same, or their behavior is same. So that is called as heredity. And variation is concerned with the forces or influences due to which no two organisms are exactly alike. So due to some of the reason, two people are not look same like, or their behavior is not like, so that is called as variation. Now let's see what is chromosomes. So in the nucleus of each cell, the DNA molecule is packaged into thread-like structures. They are called as chromosomes. So in this picture itself we can see there are thread-like structure, that is a chromosome. Now let's have a look at the picture. So each chromosome has a constriction point called the centromere, which divides the chromosome into two sections. So the small part, like the joint, that is called as the centromere, and the short arm of the chromosome is called as P arm. So this is the short arm. These two are the short arms. So these are called as the P arm. The long arm of the chromosome is called as Q arm. So the long arm. So these are called as Q arm. And the location of centromere on each chromosome gives the chromosome its characteristic shape and can be used to help describe the location of specific genes. So now let's see what are genes. So these are the basic unit of heredity that determine the unique physical, chemical and mental characteristics and traits of all individuals. So the physical, chemical and mental characteristics and traits of all individuals, or our behavior or our character, all this constitutes a gene. So now what are DNAs? So deoxyribonucleic acid, or DNA, is a nucleic acid that contains the genetic instructions used in the development and functioning of all non-living organisms. The information in DNA is stored as a code made up of 4 chemical bases. They are adenine, that is represented as A, guanine, G, cytosine, C and thymine. So DNA bases pair up with each other, A with T and C with G, to form units called base pairs. Each base is also attached to a sugar molecule and a phosphate molecule. So this you can see in the next slide. So there it is clearly given how these DNA bases are paired each, that is adenine with thymine and cytosine with guanine. Together, a base, sugar, and phosphate are called a nucleotide. Nucleotides are arranged in two long strands that form a spiral called a double helix. So this is a double helix DNA structure, and here we can see the base pairs. That is the color code. You can see the red and the yellow or orange, that is guanine and cytosine. So here it is, and again adenine and thymine, that is they are bonded together. So and that is made us, and there we can see as sugar and phosphate. They are the backbone of this DNA. Now, why these genetics are important? So the knowledge of genetics is helpful in understanding the causation of diseases, and also it helps us to understand how normal variations between individuals are brought about. So mainly it helps to identify how the diseases are originated, also how people are different, even in the same family, two brothers or two sisters or brother and sister, that character may be different. So we can understand how this variation is happening. So learning of genetics also helpful in preventing genetic disorders through genetic counseling and antenatal diagnosis, and also it helps to solve legal problems, because nowadays we know there are many issues with families, like sometimes who may not be knowing who is the child or who is the father. So in those cases also we can identify, like we can solve all the legal kind of problems. Also it also helps in preventing genetic disorders, because in the antenatal period itself, the parents will be able to identify most of the genetic disorders. So through genetic counseling, we can manage or prevent many kind of genetic disorders. Now let's go to our topic, that is genetic testing in neonates and children. I hope you understood what is genetics, what are chromosomes, what is GNA, all those things. So let's see what are the genetic tests in neonates and children. So, what is genetic testing? So, the analysis of human DNA in any of its forms or related products. Either it can be chromosomes or RNAs or proteins. Any of the product can be analyzed in order to identify the problems. Or we can use that for the genetic testing. And the genetic testing, also called DNA-based test, is used to test for genetic disorders, which involves direct examination of the DNA molecule itself. So what are the uses of genetic testing? So the genetic testing is mainly used to detect disease related to genotypes, mutations, phenotypes or karyotypes for clinical purposes. So let's see what are all these genotypes, mutations, or these things we can see in the next slide. So genotype means it is a genetic makeup, and phenotype is the physical appearance. Then mutation means a genetic change, usually one that is associated with a disease. So sometimes any mutation which happens in any chromosome, that can lead to any disease. And karyotype is a visual presentation of chromosomes. So the genetic types can be divided as mainly into two types. First one is constitutional and the second one is acquired. So the constitutional tests are for mutations that affects all cells in the body and have been there since conception. And the second type is acquired one, so that tests for changes that affect only certain cells or cell types in the body and that occurred later in life. So if we go to the Google or something, we can find many types of genetic tests, but according to the types, like mainly we are divided into two, that is constitutional and acquired. Constitutional is from the time of conception, and the second one is acquired, that is which has occurred in the later period of life. So the genetic tests for constitutional mutations are: first one is molecular test, second one is cytogenic test, and the third one is biochemical test. We will see one by one. So an example of molecular test is the analysis of DNA sequence in patient with a rare inherited disease such as muscular dystrophy. Or so in that case we can see the gene will be DMD. So it can be 1 in 3,500 male births and can be progressive muscle weakness starting in early childhood. And this kind of like these children may be by using the wheelchair by the age of 12 years and maybe death in 20s. So in the case of muscular dystrophy, how the molecular test is done means obtain the blood sample from the child and read the DNA sequence of the DMD gene and identify the mutation that caused the disease. So you may be thinking, what is the need for this information? So we'll see how it will be useful. So it can test mother, is she an unaffected carrier of the mutation? Is she at risk to have more children with this disease? So it also can test siblings of affected child. It also can offer prenatal diagnosis in mother's next pregnancy, or can offer pre-implementation of genetic diagnosis. Also can provide information about prognosis. So the second type of test is cytogenic test. So here we will use the karyotype. So to examine the chromosomal complement of an individual, including number, form, and size of the chromosome. And this is frequently used for children who present with multiple anomalies or developmental delay or autism. So here is a cytogenic test for child with autism. Here also we will obtain a blood sample from the baby, and here you can see you can look at the chromosome. So here you can see the number. So many numbers are missing and the sizes. So everything is different from a normal chromosome of the child. So again, how is this information useful? So same like the previous one, it can determine exactly which genes are involved, can test parents and siblings of affected child to see if they carry the abnormality, also can offer prenatal diagnosis in next pregnancy, or can offer a pre-implementation genetic diagnosis, or can provide information about the prognosis. So the third type of test is biochemical test. So here we analyzes the quantity of a downstream product of a gene. Example, we are not looking directly at the gene or the chromosome, but we are doing a newborn screening to check if there is any abnormality. So one of the main biochemical tests which we commonly use is the test for phenylketonuria. So as we know, phenylketonuria is an inherited metabolic disorder, and if it is untreated, it can lead to mental retardation or seizures, and it is common in, like, it affects 1 in 20,000 newborns. So here we can see in this picture itself. So for the normal conversion of phenylalanine into tyrosine, we need an enzyme which is called as phenylalanine hydroxylase. So in this case, in children with PKU, the phenylalanine hydroxylase is missing. So what happens? The phenylalanine will be or it won't be converting into tyrosine. Hence, the amount of phenylalanine will be increased in our body. So that can be found in baby's blood when we are doing the newborn screening. So how is this information useful again? Because the baby is already born and the child has already the disorder. So what can we do here? So if we identify or if we diagnose the baby in time, we can treat and avoid all clinical consequences of the diseases. So main treatment here is to restrict phenylalanine in the diet. So you can see some pictures I have ordered here, that is diet with Pepsi or some kind of tuna. So those are rich in phenylalanine. So we can ask the parents or ask the child to avoid all this kind of food. Also can test siblings of affected child to see if they are carriers for the disease. That is 1 in 70 in the general population are carriers. So we already found what are the types of constitutional tests. So that we already learned the 3 types. Now let's see what are the genetic tests for acquired mutations. So here also we can see molecular tests and cytogenic tests. So these are tests for changes that affect only certain cells or cell types in the body that occurred later in the life. So molecular tests for acquired disease. So CRAS is a gene test on tumor tissue from patients with colorectal cancer. So what we do here is obtain tumor from the patient and extract DNA, treat with enzyme that allows visualization of the mutation. So, how is this information useful? So, patients whose colon tumors do not have a CRAS mutation are much more likely to respond to cetuximab therapy, and also identifies patients most likely to benefit from specific therapies. Also, it allows choices of alternative therapies and saves time and money for patients unlikely to respond. Again, cytogenic tests for acquired disease example is that is HER2 new gene amplification in breast cancer. So this occurs early in oncogenesis and can see up to 1 in 3 of breast cancers that are associated with poor prognosis and response to Herceptin treatment, but does not respond to tamoxifen treatment. So how is this information useful? So it assists in selection of patients for chemotherapy and which therapy to use, predicts response to adjuvant therapy, and also it increases survival, allows choice of alternative therapies, and saves time and money for patients unlikely to respond. And genetic testing for specific conditions. So what we do is take a sample, that is blood or amniotic fluid or mouth swab, and use staining of chromosomes to locate any chromosome abnormalities, or use matching DNA sequences or antibodies to detect gene abnormalities. So again, genetic testing in neonates, which is performed to diagnose a disorder or screen for a potential pathological condition. It can be done by testing newborn blood sample, as we already have seen for the diagnosis of PKU, and also we can see galactosemia or trisomy. Also, the neural tube defects are common in some of the children. So, example, spina bifida occulta, that is multifactorial deficiency of folic acid in maternal diet, positive family history, etc. So, in those cases, we can do screening tests by maternal serum alpha fetoprotein or ultrasound or by CT scan. Again, in order to identify errors in metabolism, that is PKU, blood and urine test can be done. Also, congenital hypothyroidism can be identified by neonatal screening for T4, TSH, or both. Galactosemia can be identified by screening of urine or serum for galactose. Then, cystic fibrosis. And again, trisomies 13, 18, and 21, that can be identified by amniocentesis, chorionic villi sampling, USG, or karyotyping. Again, let's see what are the genetic testing and screening which can be used in children. So the main approaches are category 1, disorders requiring immediate treatment or prevention or surveillance. That is phenylketonuria. As we already said, the dietary management can be done. And again, multiple endocrine neoplasms. So that is life-preserving thyroidectomies can be done. And in category 2, testing asymptomatic carrier to define reproductive risk for future. So they are sex-linked disorders, example hemophilia or muscular dystrophy, and again autosomal recessive disorders like cystic fibrosis, sickle cell diseases. And in category 3, we can see testing healthy young person with predisposition to develop disease, that if the family has breast cancer or hereditary non-polyposis colon cancer. So in those cases, we can avoid environmental dietary factors. And in the category 4, testing healthy young person who has polycystic kidneys or Parkinson's diseases. So whichever the genetic testing we are doing, the ethical concerns are very important. So here confidentiality should be maintained throughout the procedure. And comes to the nursing aspects. So mainly we need to assist with the testing procedures, support the client and family, appropriate education and clarification should be given to the family, and also support for the family who chooses therapeutic abortion before, during, and after the procedure. And also should be able to explain the complete genetic information to the family in a simple way, must take a thorough family history, information about exposure and lifestyle, information about the affected and unaffected conditions. So comes to the summary. Constitutional genetic tests impact patients with inherited disorders and their families by providing information about diagnosis, prognosis, treatment, and informing reproductive decisions. Genetic tests for acquired diseases impact patients with diseases like cancer, provide information about diagnosis and prognosis, and inform treatment decisions. Genetic testing impacts many phases of our lives and will do so even more in the future. Thank you.